So, hello friends, today my video is about physiology of hearing. My previous two videos were on external ear and middle ear. So this one was bound to be on inner ear. However, I have clubbed two things together, that is physiology of hearing and anatomy of inner ear. So it becomes easy for you to correlate both the things and gain better knowledge over the topic. So as we all know, ear has got two functions. The first one is, as all we know, it is hearing, and second one is balance. We all need to maintain balance while doing various activities in our day-to-day life, so that balance comes from your inner ear. Have you ever noticed you feel giddy at times when you have been through a merry-go-round or something? So that giddiness is produced because this balance is not maintained. Okay, so the scientific term for that condition is vertigo. Vertigo is it has got many other clinical etiologies related to it as well, but for now, uh, balance is maintained by inner ear. Okay, so for hearing, the structures which are important, these are rhinous membrane and basilar membrane. Both of them are present in your inner ear. The details I'll be explaining in the diagram below. And for balance, it is scala vestibuli, scala media, scala tympani. Okay. So diagrammatically, I'll represent this. So this is your scala vestibuli. This is your scala media. This is your scala tympani. So all three of them have got fluid present in them. So this is a bony cage called as cochlea, into which these three compartments: scala vestibuli, scala media and scala tympani. So the fluid which is present in scala vestibuli is and scala tympani is perilymph, while the fluid that is present in scala media is endolymph. Okay, so this is perilymph, this has got endolymph. So what is the difference between this perilymph and endolymph? Perilymph has got more of sodium ion concentration. More sodium is present over here in perilymph, while endolymph has got potassium, lots and lots of potassium ion concentration. The concentration of potassium ions is more in scala media. So these, as you may have learned in physiology before, how is action potential generated? So what do we need for action potential is basically sodium ions, but here the thing is different. For this hearing purpose, the endolymphatic potential is developed, and this endolymphatic potential is produced by your potassium ions, which is 80 millivolts. Okay, so scala media has got your hearing receptors. This is the membrane which secretes your endolymph. This is scala vestibuli, media, tympani, VMT. Okay, VMT. Very minute things. So this is a mnemonic which I'm giving you, very minute things, because you always get confused which one is where. So the sequence is from top to bow, very minute things, that is vestibuli, media, tympani. Okay, scala media. Scala media consists of your organ of Corti, which is the main hearing organ. So where is your organ of Corti present? It is present in your scala media. Now what it consists of? So this is the organ of Corti. This is a schematic representation. What do you have in here? So you have your hair cells, inner hair cells and outer hair cells. For what are these needed? These are needed for your hearing purpose. And these are your diator cells. Do dieting? No. So these diator cells are needed, and supporting cells for inner and outer hair cells. So which are the supporting cells in your nervous system? Yes, these are neuroglia. Then you have astrocytes in your brain. So we have these kind of supporting cells everywhere in our body. So In the organ of Corti, these are known as diator cells. Okay, so outer hair cells are more prone to damage by drugs. And which are those drugs and what do they do exactly? So details of this are to be learned in ENT, but one drug I'll tell you which we consume a lot. So that drug is aminoglycosides. Okay, then the other drugs are furosemide, uh, then paclitaxel, cisplatin, all those chemotherapy drugs. So they cause ototoxicity. Simple thing. Again, this term is not new. Ototoxicity. Oto means ear toxicity. Whatever is causing toxicity in your ears. So that damages your hair cells. And what will happen if your hair cells are damaged? You won't be able to hear things. Okay. So the receptors are innervated by your eighth nerve, and which is the eighth nerve? Auditory nerve. Okay, and then they are carried to the spiral ganglion. The auditory pathway I'll be telling in the next video, but for now the eighth nerve is carried to the spiral ganglion. Now, are these inner and outer hair cells the same? Well, in the diagram they may appear same, but they are not. Inner hair cells are very less in number, while outer hair cells are numerous. Inner hair cells are highly innervated, while outer hair cells have less innervation. Okay, and which is the tallest hair cell? So, the is tallest hair cell is Kinocilium. Okay, so the process, exactly what happens in the physiology of hearing, I'll be telling in the next video, because this video is getting a little longer. Thanks for listening. Do revise this physiology of hearing, as it is very important for your first year as well as for your third year. Okay, thanks for listening.